All right, good afternoon, everybody. Thank you for coming. Welcome to the seminar series, Faculty of Agricultural and Food Sciences. My name is Nazim Chichek. I'm the Associate Dean Research for the faculty. And it's really my pleasure to introduce the speaker today and run the Q&A session. But before I even get there, I wanted to make a quick announcement. February is Black History Month in Canada, and we have a special seminar and a panel discussion happening next Tuesday in this room between 2:30 and 4:30. We have 5 speakers on the panel. 3 of them are our faculty members. One is a student and the other one is an ally. And what's going to be fun is we're going to have some different foods here for a light lunch, some presentation, and hopefully a robust discussion. So I'd urge you to all make time for that. Again, 2:30 to 4:30, February 4th, next Tuesday in this room. All right, without further ado, let me introduce today's speaker. It's Jason Gibbs from the Department of Entomology, and Dr. Gibbs received his Bachelor's of Science in Biological Sciences from the University of Toronto in 2002, and his MSc in Botany from the same school in 2005, his PhD in Biological Sciences from York University in 2009. Followed that up with a postdoc at York between 2009 and 10, and then moved to Cornell to continue his work there between 2010 and 13, and became a research associate at Michigan State University between 2013 and 16. Then we were able to attract him to the U of M, where he started his assistant professor position in December of 2016 in insect taxonomy in the Department of Entomology. Jason is also the curator of the J.B. Wallace R.E. Ruffley Museum of Entomology, one of the largest insect collections in Western Canada. He was promoted to associate professor in 2022. His research focuses on wild bee taxonomy and biodiversity, and he has co-authored over 100 scientific papers. Also, he currently has 4 species of bee named after him. So when you're swatting away a bee, it might be one of the Gibbsies, so be careful. Without further ado, Jason, take it away.
Thank you very much. It's highly unlikely you'll see any of the bees that are named after me unless you go, there's one at Spruce Woods, but most of them are exotic. So today I'd like to talk about the bees that no one knew were there and no one wanted. And this is the part of the talk where normally I would tell you why bees are really important, but I couldn't be bothered. So I just asked an AI to give the response of why are bees good. And this is the headers for what how they explained it. So pollination, bees provide that to both crops and wildflowers. They're an important part of biodiversity. Certain species make honey, very few. They can be important ecological indicators. There's some data that suggests that bees will respond to environmental changes more quickly than other species. Economic value, largely from the pollination and the honey production, and cultural significance. So as far as insects go, people like bees as a general rule. Although when I first started studying bees, usually when someone saw me with an insect net, they asked what I was doing, and I would say I'm collecting bees, and they would usually ask me about killer bees. But that changed dramatically in around 2007 when there was colony collapse disorder in honeybees. Now everyone likes bees, which is good. So the part of my talk I'll cover kind of 2 different aspects. The bees that no one knew were there is mostly about the distribution of species and learning more about where species are, and a little bit about undescribed species. And then the bees that no one wanted are really a group of bees that I have spent the last 15, 20 years studying that people really hate, which is good. But I want to quiz people. I presented basically a similar slide 8 years ago when I gave a faculty seminar, so I wanted to see if anyone remembered, and also for the new people. So how many bees are here? If you're watching online, you can put it in the comments. But if you're in the room, how many bees do you see in this picture? Feel free to shout out an answer. Some people probably know the answer to this if you're in my lab. Any guesses? 3, pretty good. 1, good. 11. Someone's playing devil's advocate. Okay, lots of answers. I always say 2. The answer to this is 2. So there's 1 there. It's a trick. And then the other one is in the bottom left. So all of the rest are flies. So even flies recognize that bees and wasps are the best kinds of insects, and they try to mimic them as often as they can. So what are bees? There's 2 insects here. One of them is a bee, one of them is a wasp. The one on the left belongs to a group called digger wasps. They like to make nests in the ground, and they hunt prey. They paralyze them with their sting, and then they lay their eggs on them. Bees are basically just a vegetarian digger wasps. Instead of using insects for food for their larvae, they use pollen as a protein source. So this is a phylogenetic relationship showing the evolutionary patterns within digger wasps, and bees are just kind of nested in there up at the top. Even though they're just a small portion of this tree, they're actually twice as diverse as all the digger wasps combined. And that one in the upper right is just the closest relative, we think, of bees currently. And we know very little about that group of wasps other than one species hunts thrips. And so the sort of made up story that people speculate is, there may have been some little wasps and they were hunting thrips on flowers and got a little bit of pollen in their they fed their larvae pollen sort of incidentally with their prey, and then somewhere along the lines, gradually they became pollen specialists. But besides just being vegetarian wasps, bees are prettier than and cuter than wasps as a general rule. So this is Calides wilmati. It's, as far as I know, it's only known from Canada in Spruce Woods Provincial Park. It's a specialist on dahlia prairie clovers. This is Dahlia villosa, so it's a rare plant, a rare bee on a rare plant. And it lives solitarily, nests in the ground, quite different. So bees are actually pretty diverse. So there's more species of bees on the planet than there are mammals or birds or reptiles. And honeybees and bumblebees, the ones people are usually familiar with, make up a minuscule proportion of that diversity. You can't even see honeybees in the bar graph. There's about 21,000 species of bee globally. And when I was making this chart, I thought, well, I should probably figure out what the actual numbers of mammals, birds, and reptiles are. So I asked AI again, and I was ecstatic to find out there's almost 11,000 reptiles in Canada, including 24 crocodiles. So that was news to me. But this also made me very happy to know that I'm not going to be replaced as a taxonomist by a robot anytime soon. So a lot of the pollination services we credit honeybees with is often other insects. So this is just a summary of different studies from around the world, and the proportion of the little pie charts in orange is how many visitors were honeybees, and green is other things. And so oftentimes we sort of give credit to honeybees for pollination, even though other insects are actually doing a lot of the work. So the logic kind of goes, we have an apple orchard, we need it to be pollinated, we put a honeybee hive nearby, we get apples, and we say, oh, all that apples were thanks to the honeybees. But if you actually took the honeybee hive away, you'd still get apples. Most bees are nothing like a honeybee, so that's a honeybee up top. And then the little small one is the group of bees that I'll talk a little bit about. These are called sweat bees. They belong to the family Halictidae, and that's in the genus Lasioglossum. They're adorable little bees. But these are the bees that nobody wanted. So they've been called monotonous and uninteresting bees, the despair of taxonomists, morphologically monotonous by the world expert on bees, Charles Michener. Ron McGinley was a sweat bee taxonomist. He called them an acquired taste. Jerry Rosen, who is a retired curator at the American Museum of Natural History, said we should kill more of them. And recently, as recent as last week, Sophie Cardinal, who curates the bees of the Canadian National Collection, was very specific in her email that she really dislikes Lasioglossum. I've called them a nightmare taxon in print. I used to have a lot more hair. It's all been pulled out. But actually, when you get used to them, they're actually pretty, when you look at them under the microscope, they can be quite attractive. They can be quite diverse. These are all the genus Lasioglossum. And so I want to talk a little bit about why I sort of focused on this group that nobody else likes. One is they're very species rich. So there's about 4,400 species in the family. Over 1,800 of those are in the genus Lasioglossum, and we're finding more of those all the time. So this about 8% of all bees are in this genus. About 400 in the genus in North America and 62 in Manitoba. So that's quite large. So there's about 5,200 species in North America. So again, a fairly large proportion of the total diversity. And they get their name sweat bee honestly. So this is actually, you can sort of see the mouth parts of the bee. They sort of fold up underneath the head. So they're extended. This is on my thumb. And she is drinking the sweat off my thumb. And they presumably do that to get certain sort of micronutrients, salts that they find difficult in the environment. So if you go outside on a really, really hot day, there's a decent chance a sweat bee will land on you. And sometimes some of the best sweat bee collecting I've had is collecting off other people with their permission. Lasioglossum are incredibly widespread. So they occur in virtually every terrestrial habitat. Usually if you see a distribution like this with points in the middle of the ocean, someone got the GPS coordinates wrong. But in fact, they actually do occur on all these islands. They're really good at finding their way into novel habitats. Some of them are quite widespread. This is Lasioglossum lavissimum. If you ever see a sweat bee on a dandelion in Winnipeg, it's probably this one. They're widespread across North America. In the bottom right is just records that we have in the collection from Manitoba of this species. Other sweat bees, though, are really restricted. So this is a species that's only known from a few locations in Alberta and Saskatchewan. The black squares are where they've actually been collected, and then the heat map around that is predicted suitable habitat based on climate models. And that's what the bee actually looks like. It's quite pretty. So this was a new species that I described with one of my students a few years ago. And these are the habitats that it's been found in. So it's very specialized on sand dune habitats. Spruce Woods Provincial Park also has sand dune habitats similar to this, and we've sampled that extensively, and we've never found this bee there. So it has a really restricted range. Sweat bees are also really abundant, and this is part of the reason why people hate them. So if you go out and collect bees, about 30 to 70% of them are likely to be sweat bees. And because they're sort of morphologically monotonous, people are forced to sort of deal with them. And in the past, people would usually just say in their papers, we caught 1,500 Lasioglossum and we didn't bother to identify them to species. As pollinators, it really varies. So here's a couple of images of them not pollinating. The one on the top is just nectar robbing off that plant. The one on the bottom is collecting lots of pollen, but she's on a lily and she can do that without getting anywhere near the stigma. So sometimes they're robbing, but in other cases, they're the only or primary visitors of certain plants. And they're very good at sort of remaining in agricultural systems. So they're very tolerant of disturbance, unlike a lot of other bees. So part of my job as a taxonomist is try to solve this question of how we tell them apart. And I've been relying a lot on some excellent graduate students. We spent a lot of time looking through microscopes, trying to find subtle characters and taking measurements, and sort of publishing papers to try to help resolve the sort of taxonomic impediment that limits other bee biologists from doing the work that they want to do. So these are just some recent papers by students. Sometimes when you have a morphologically challenging group, one solution would be to use molecular data. So DNA barcoding is a traditional way of doing that. And it kind of relies on the idea that the amount of variation within a species will be fairly narrow, versus the amount of variation between species, which will be relatively large. And if there's sort of a gap between those two distributions, you can sort of easily sort of separate species into one group or the other. For Lasioglossum, it doesn't really work. It overlaps extensively. And so you can't just barcode your way out of it necessarily. This is based on a data set of 106 species that were recognized kind of traditionally. And molecular sort of approaches suggested there may be 90 species. So there's some disagreement. It could be, so these are, those, 90 species were sort of a so-called, given unique identifiers called barcode index numbers. And you can get kind of a one-to-one correspondence where taxonomy and these barcode index numbers match. You could have a situation where the barcode index numbers merge traditionally recognized species, or divide it into more than one, or some sort of complicated mixture of those two scenarios. And in this plot is kind of showing the sort of the distance between species on the x-axis and the distance between species on the y-axis. Everything in orange is really good, no problems, everything agrees. Over here, we may have some undescribed diversity that we need to deal with. But in green, on these green triangles, that's all the areas where the DNA barcode suggests that they're not multiple species, even though as taxonomists we recognize them. So this might be suggesting that I'm a bad taxonomist. And I want to assure you that I'm not at least the worst. So this is two different species. That's a little fuzzy, but this individual has a very smooth thorax with distinct punctures. This other one has a really coarsely sculptured with all these deep wrinkles on the side. This is called obscurum. This is called atwoodi. And this is a summary of the DNA barcode data. So the length of the branches this way represents how much sequence divergence there is. And if you look at the top, there's 0. The DNA barcodes show no sequence divergence between these two individuals. And because these are so morphologically monotonous kind of bees, that's about as different as you can get. So if those are the same species, we're in huge heaps of trouble. And there's various reasons why this might be the case. One of them might just be that they diverged really recently. And so this is just sort of a theoretical kind of map of a phylogeny with sort of individuals and sequence very different sort of haplotypes sort of represented within each species and each lineage. And as they separate into distinct species, depending on how much time has passed, you can either get these sort of nice discrete clusters, or you can get this really messy sort of story where you can't distinguish things. And this might be the case in Lasioglossum. They're also known to have a lot of these endosymbiotic bacteria that are maternally inherited, and that might sort of, as those bacteria become infected in different populations, they might obscure the amount of variation that has accumulated over time. So there's all kinds of reasons why the barcodes don't work. So one solution to this is to get more data. And so we've been exploring the use of something called ultra-conserved elements. And these are parts of the genome that are really deeply conserved, so you can see the same sequence across distantly related species, which is not very useful, except as you move away from those conserved elements, the sequence variation increases. So this x-axis is just the distance from the core ultra-conserved element, and this is the amount of sort of variation that accumulates. So we can actually use the ultra-conserved elements to find parts of the genome that are shared across different species, but then sequence the flanking regions to get informative data that we can use to discriminate populations. Usually this is used for phylogenetics, but there's some data that suggests that this might be useful for discriminating at the species level. So basically, what we do is you have these sort of ultra-conserved elements. We take our DNA, we fragment it. You get these UCEs basically as commercial baits. You attach them to magnetic beads. You basically go fishing into your genomic DNA, and it sucks up the ultra-conserved elements. And then you can sequence it with next-generation sequencing. So preliminarily, this is this problematic group that barcodes can't discriminate. Preliminary data suggests it might be sort of recognizing species. So that's one sort of species there, sort of matches, that's another one. That's an undescribed species that was identified morphologically and it's discrete. But there's other areas that are still a mess. And this may be because we don't know how to identify them morphologically yet. So the summary of this part of the talk is you can't handle the Lasioglossum. They're a challenge. But we're working on it, and we hope to make further progress. Part of the reason why we want to is because they're such an interesting group of bees behaviorally. So they have more social complexity than any other anything. So everything from solitary species to communal nesting, where they're basically sharing a nest entrance, division of labor between members of the same generation, division of labor between members of different generations, brood parasites. Some of them nest in the ground, some of them nest in wood. That's evolved independently. Some of them are nocturnal or crepuscular. That's evolved independently multiple times. So insects have compound eyes, but they also have simple eyes, these sort of simple ocelli. And especially the one in the upper right, those ocelli are greatly enlarged because that bee is flying at night. These are 3 different lineages, one from North America, one from Australia, and one from Eurasia. So that's sort of arisen multiple times. And one issue where sort of taxonomy and behavior sort of meet is that you can't tell what they're doing just by looking at them. So these are all similar looking species, but they have strikingly different behavioral nesting behaviors. So being able to identify them then allows us to sort of better understand what these bees are doing in the environment. In eusocial species, the traditional way is that they mate in the fall, and then only the mated female overwinters. And then she'll initiate a new nest, and then her first brood is all females. And then only later in the summer does she make reproductive males and females to sort of continue that cycle. But I just wanted to mention an especially interesting species called Lasioglossum marginatum, which occurs in Europe. In this case, the female overwinters. She's mated in the fall. She initiates a nest, makes a brood of workers, and then they close the nest up. And the workers overwinter until the next year. And then they open up the nest in the spring. They go out and forage. She makes another brood of workers, and then they close the nest up again. And then they overwinter all collectively as a colony. And then the next spring, they open up the nest, and they all emerge, they forage for more pollen, make another set of workers, and then close the nest up again. And they do this 5 to 6 years in a row. So it takes 5 to 6 years for them to actually go and make new reproductive males and females. Why they do this, I have no idea. How that is an evolutionarily successful strategy. You're putting all your eggs in a 5 to 6 year long basket. But they do it, and that makes them cool bees. And there's also a number of brood parasites. So these are bees, Lasioglossum, that go into the nests of other bees and live there and don't collect pollen and don't build nests. They just lay their eggs in the nests of other species. And that itself has arisen multiple times. And so one of the things that we like in systematics and taxonomy is creating phylogenies. And halictid bees of all the sort of social insects seem to have evolved social behavior more recently than others. And it makes them a really good test case for understanding the origins and maintenance of social behavior. And it was in sweat bees that people sort of first kind of realized that the evolution of social behavior is not a one-way street. So usually people thought that colonies would get increasingly complex until you evolved eusociality with queens and workers, and that was kind of an end point. That was, there was no going back. But in sweat bees, people found with phylogenies that in fact you can reverse and go from a highly social colony with queens and workers back to a solitary one. So this is just showing the evolutionary relationships, and people just mapped on the different behaviors, whether they're solitary or eusocial, onto the tree, and they basically kind of work backwards to the common ancestors to figure out what they were like. And discovered that, in fact, you have these social lineages that sort of reverted back to solitary behavior. And this is why the sweat bees are called the most interesting bees from the perspective of social evolution. And I was participating in some of these studies, and so this is sort of showing the likelihood of a social common ancestor at sort of different points. And we've been using the ultra-conserved elements to make increasingly better phylogenies of sweat bees so that we can understand the development of social behavior in greater detail. Okay. Now I want to talk a little bit about the bees that no one knew were there. So when I came to Manitoba in 2016, there had been a recently published checklist of the bees of Canada in 2015, and it listed 250 some species for Manitoba. And over the course of a few years, with a number of students, some of whom are in this room, we came up with a list of 392. So that's a 60-some% increase in the number of species known from the province. This is data pulled from the museum's database, sort of mapped onto the province. And so there's a pretty strong bias in sort of sampling for bees. We've sampled quite a bit in southern Manitoba, but relatively little in northern Manitoba. Some of the ones that we do have up near the Gillam are just, the lucky, fortunate efforts of like a single person who was nice enough to go out and put out some bee bowls. They're also in this room, Todd Lawton, thank you very much. But in our sort of collections and sort of going through historical material, we found all kinds of interesting bees that no one knew were in Canada even. So these are nomad bees. They're brood parasites. People would usually sort of think of them as being wasps if they saw them. They are wasps in a sort of broader evolutionary sense, just vegetarian ones. But each one of these species had never been documented for Manitoba. Some of them are incredibly rare and are only known from a few individuals. So quite cool. This is another group. We call these the smiley face bees because of the hair pattern on the thorax. There's actually two different genera here. One of them had never been recorded in Manitoba, so we were finding an entirely different genera that people hadn't recognized that were here. And this is the infamous Epiolus gibbsi that was collected in Spruce Woods. It was either me or my wife, but I'll take credit. She was with me at the time. And so to understand and document bee diversity in the province, we use a variety of methods. Some of them are passive sampling. This is just sort of from my student Thelina's past summer project, using different colored bowls with soapy water. The bees kind of are attracted and drown, and then we pin them and make them famous later. And also just netting. We go out and actively net bees from flowers, and that helps us understand the floral interactions, the plant pollinator interactions between the bees in a way that we wouldn't otherwise get. So that's Thelina in the upper right, and that's Reed, who's also a former graduate student. So usually these graduate student projects are sort of focused on a specific question. In this case, my former student Emily was trying to look at the relationships between the effects of landscape on bee communities and how increasing amounts of agriculture affected bee communities. She sampled across southern Manitoba. And this is just some of the summary slides from her thesis. But in the upper left, you have bee abundance. Upper right, you have bee species richness. And both of those are negatively affected by the amount of cereal crop cover in the landscape. Bees don't like wheat and rye and things like that. The other two graphs are measuring something called functional dispersion, where basically Emily was looking at the different life history traits of those species and trying to understand how what kind of trait diversity was present in the communities. And those have different responses to kind of landscape. Bees kind of like edges. They like being at the edges of habitats because there's a lot of floral diversity there, presumably, and nesting habitat. So one of the other things we've been working on in the lab is trying to increase the amount of attractive edge habitat for bees. So we had an organic cluster funded project a few years back with Yvonne Lawley, Alejandro Costamagna, and Rob Goulden. And we put in these pollinator habitats next to fields to sort of measure the effects they had on bees, but also other beneficial insects. And this is the picture I always show because this is the nicest they ever looked. Turns out it's actually hard to establish this kind of habitat. And so one of the things that we're doing in this new Sustainable Agricultural Research Initiative project is trying to improve understanding of how to make these effective. But my student, Mike Killewald, basically found in the first year there's this big kind of strong effect that these floral strips increased the number of bees that were present compared to controls and some natural sites. And in another sort of paper that's currently in revision, he sort of showed that effect kind of maintained. It kind of drops off at the end, and that might be because of the declining quality of the strips over time. We had a drought and then floods. It was not good. The other thing we're sort of exploring in the lab currently is trying to incorporate phylogenetic diversity into our studies of bees. So this is, again, is using those ultra-conserved elements to create phylogenies of all the bees that we're collecting in Manitoba and the prairie provinces. And the reason we want to do that is because bees don't respond to environmental changes the same. And part of the reason why is because of their different traits. Some of them are social, some of them are solitary, some of them are big, some of them are large, some of them specialize on particular plants, some of them are very generalists. And so we can measure that, and that's what Emily was doing with that functional dispersion metric. But there's a lot of things that are under the hood that we're not measuring, we don't see, that might be influencing biochemical, physiological processes that are evolutionarily conserved. And so we want to try to get at that by measuring phylogenetic diversity. And that's kind of ongoing work with Thelina, funded through Genome Canada. And there's some precedent for suggesting this might be important. So there was a study in apple orchards in New York that was measuring the proportion of agricultural cover on traditional measurements like a bee abundance and species richness, but also phylogenetic diversity. And it was phylogenetic diversity that was the best predictor of the actual outcomes on agricultural production. So yield was influenced by phylogenetic diversity more than other things. So the other thing that we're kind of trying to understand is how bees and the sort of the economic or ecological services they provide are distributed in the landscape. So this is another sort of output from our database, kind of showing different sort of collection localities of where we have bees. So it's not just a difference between the north and the south, but you can probably notice that there's certain areas of the province that we have not done a very good job of sampling. And often these are the areas where agriculture is strongest. We think that bees don't like highly disturbed agricultural systems, at least some bees don't. And we like bees in the lab, so we like to go places where we think there's going to be lots of bees. So we tend to collect in places like Spruce Woods or in sandy lands. Winnipeg just has a lot of bees just because it's Winnipeg, and this is where we all live. Things like the Tallgrass Prairie Preserve has lots of records. But we actually don't know very much about a lot of the province. And so another thing that we're hoping to do in the lab, I have a new PhD student starting in May, is to try to do some more kind of spatial analysis kind of measurements to try to understand a little bit more about where bees are. And hopefully we'll try to fill in some of these gaps. And this kind of relates to, I'm going to show this. This is kind of a complicated slide, but I'll walk you through it. This was a paper that came out for the United States from a few years ago. And they were using crop data layers for the country to try to measure where bees were likely to be diverse or sparse. And this is largely based on expert opinion. So in the upper left map is basically represents, the blue represents where there should be a lot of bee diversity, and the yellow represents where we think there'll be very little bee diversity. But an interesting thing about this paper was that the second map in pink and green represents the uncertainty that people had in those responses. And there's pretty close correspondence. We think we know what's bad for bees, but we don't have a very good idea of what's good for bees. Partially, I think it's because the things that we think are good for bees are habitats that are very diverse and complex, and so we don't have a good appreciation for how bees are responding to that complexity. So this might actually be good because we actually have the most actual empirical data from those diverse habitats in Manitoba, and hopefully we can use our real data to do similar sort of things, but not just based on expert opinion of people saying, I think that's a good habitat, or that's a bad habitat, but actually have data to support it. This third map was also basically the likelihood of change in those habitats, so whether or not things were going to get worse for bees or better for bees. Usually things in red are getting worse, so habitat is getting worse for bees, we think. And then the upper plot is basically where things are getting worse or where change is happening versus where pollination is actually needed for bees. So those are sort of major crop growing areas where we think bees are important and would be beneficial, but where there's been a lot of change over time. And again, there's a map for basically how uncertain we are in that. And then this last map is basically where these sort of red circles are basically where things are going badly and we have an increasing amount of need for pollinators, but pollination services we think are declining. So we want to know more about these sort of patterns, but in a Manitoban context. One thing we're sort of interested in exploring is the use of drone technology to sort of get a better sense of the habitats that are present for bees locally. So you can use drones to sort of take kind of vegetation kind of measurements, essentially. There's been some research focused on things like surveying for weeds. So if you can fly a drone over a crop, identify the weeds, and then use that to have more targeted sort of spraying of herbicides. But we basically want to use similar technology, but to identify plants that are going to be good for bees in the landscape. So this is kind of the weed-specific model of using drones to create these maps and then have sort of selective treatment. And one of the benefits of that is increased biodiversity. So that's what we're kind of interested in, increasing biodiversity for bees. So we want to go into these sort of landscapes and measure them and then quantify what the bees are actually present so that we can sort of determine what habitats people should be trying to retain, because those are going to have beneficial benefits for these insects and potentially other predators and other things that people are out there. And there's some precedent for this. So this, I took this out of a paper that used a similar approach that came out a couple of years ago. And basically, they were comparing these sort of drone survey techniques to boots on the ground, visual observations of plants and flowers for bees. And the drones, there's different models for the drones, but the one in blue has the highest R squared, so it was the best predictor of the data, was actually better than the visual observations. And that may be in part because the drones aren't restricted in the same way that we are. They're able to visualize a greater area, but they're also sort of seeing the world in a kind of bee's eye view. They can kind of fly over and see the things that bees would see. So we can potentially use that to sort of get an assessment for the local sort of conditions and then use that to predict bee diversity. That's what we're hoping for. Okay, so just to sort of summarize, bee diversity is complex and poorly understood, but we're trying, one of the focus of the lab is trying to sort out that complexity. Landscape and habitat affect bee diversity, but they do it in different ways and in ways that are complex and we don't fully understand. But that, newer tools like phylogenomics, spatial mapping, drones have the potential to sort of fill in some of these gaps and improve our understanding of bee diversity. And lastly, there's too many Lasioglossum, but that's good for me. It's gives me lots to do. There's too many people to acknowledge, especially for the taxonomic work. I have long overdue loans from various museums and collections. So I really thank everyone who's patient with that. Various graduate students in the lab have made major contributions to this, and people working in the lab in the molecular side as well. Various funding agencies, continuing projects. So thank you very much.
Thanks, Jason, for this excellent presentation. And I failed to mention this year's theme is digital ag. So now you see the connection, right, with the data work that he's presented, certainly the spatial mapping and the drone work. So that's part of the reason why we asked Jason to present in this year's seminar. All right, so time for some questions. So fire away.
So how many complete genome assemblies do you think there are available for the sweat bees?
So the question was, how many complete genome assemblies are there for sweat bees? Few, but more and more. So there were a few full genome assemblies sort of in different parts of the halictid phylogeny. There was a Euphoria, which I was actually helped collect the specimens for that, and a few of the social bees. And there has been, especially in the States, an effort to get increasing numbers of full genomes for bees. And there's also a bit of an effort in Canada that was being run out of UBC, where they're trying to get full genomes of a number of different insects. And so we have some bee samples that we collected here in Manitoba that we sent to UBC to get, or the Genome BC, what it was called, to get full genome. So those are in process. So we'll have some more. Maybe, a few years from now, we'll have hundreds of genomes for bees, but right now it's still pretty few.
Any other questions for Jason? Go ahead.
Jason, have you tried to project from the number of species you've collected so far in the province, how many more there are, what proportion you might already have?
Yeah, so the question was, have we tried to project from what we currently know about how many species there are to try to, like a kind of a rarefaction or a species accumulation kind of approach to figure out how many species there actually are. I can say that almost immediately after that paper was published, I found additional species that students had collected. So there is actually more species now than there were, and Todd Lawton, who was here somewhere, he was up in Gillam collecting, and he found a species of bee which I've never seen before. I don't have any idea what it is. It might be an undescribed species, and so that number is going to accumulate. But I've never actually tried a species accumulation curve of like numbers of samples to see like where we are on that. It would be an interesting project. There's about 98,000 specimen records digitized for the province, and I imagine several thousand more that I still have to deal with, and more coming in all the time because students keep collecting bees, which is awesome. But yeah, so I'm hoping that at least we'll cross the 400 path point, arbitrarily number.
I think I saw a hand go up. Yeah, go ahead.
In recent years, I've noticed that bee balls have become sort of everywhere.
Mm-hmm.
And I'm wondering, this is yet another sort of biased sampling method.
Mm-hmm.
What don't bee balls normally get?
Okay, yeah, so the question was, people are increasingly using bee bowls, but are they biased and what are they not collecting? Definitely true. So bee bowls, fortunately for my career, are very good at catching Lasioglossum, but they tend to be biased against larger bodied bees. And so oftentimes we try to include at least net collecting. Netting seems to have do a really good job of getting a good measure of the diversity of bees from the number of specimens you collect. But it's labor intensive and has its own biases and differences between collectors. So we try to use multiple methods to try to get the maximum sort of sense of the community. We have other traps that we sometimes use, blue vein traps, and they're super biased in an entirely different way than pan traps are biased. And so if you actually do like a community composition analysis of nets and blue veins and bowls, you get completely different communities from the same location. But we hopefully, if we just collect them all, we'll have
So that's what your plan is for this sort of hope for more systematic study of the distribution of bees across southern Manitoba.
Yeah, it becomes really tricky, especially when you have using historical data, because people collected bees in very different ways than we collect them today. And so, we can't necessarily just say how many bees were collected, 20 years ago, like when Terry was collecting lots. It's very difficult to make the comparison to that data other than like a presence absence kind of approach. One thing that we've been trying to implement since I got here was like a fairly standardized way of net collecting across student projects. So be a standard sort of distance, a standard time, so that we can compare Mike's project to Emily's project to Sid and Kyle's lab used a similar approach. So hopefully over when I retire, we'll have a big data set of like multiple different student projects that we can combine.
Just a warning, if you're
It won't work.
You won't retire.
Retire in the sense that the university stops paying me, but I actually expect to be much more productive when I retire, if I'm alive still.
Can I ask you something? I was intrigued by the bee's eyes view of the drone. And I wondered, the data shows the drones are doing a better job, it seems, than even the human sampling or identification. How does a bee see? And does the drone technology simulate that? I don't know if you know the answer. It's just, I see a bunch of eyes on them, right? And different ways of nighttime versus daytime. You mentioned this already. Is there a correlation between the technology of the drone and how a bee sees things?
Yeah. So the question was whether how does the drones kind of view the world differ from the way bees view the world, and how do bees view the world? And they see it similar to us, but their color vision is sort of bent more towards the UV. So you may have seen images of like with the UV filters on cameras where what looks like just a plain white flower actually has all these bands that are sort of drawing the bee towards the center of the flower. So that's an interesting question of whether or not it would be the drones would have better success if we had those sorts of UV sort of we're targeting those sorts of UV wavelengths. But I think partially what I always people I'll sometimes have students who will do these sorts of observation transects to try to better explain why some sites had more bees than others. But you're doing it on such a small scale, and the bees can fly so far that maybe just on the other side of that shelter belt, there's some bush that's like got tons of blooms going, and there's just covered in bees, and you're just missing that. But hopefully with a drone, you'll just get a sense of the whole area that's covered by bees.
Very interesting. Any other questions? Yeah, Mark?
Thank you, Jason. I'm observing all of your research supporters in the screen behind you, and it's really hopeful actually to me to see such a broad constituency coming together to support your group. So I guess I wonder if you would care to comment on if there's any sort of what the general enthusiasm for biodiversity is within those groups, if there's any sort of unifying themes, and does it make you hopeful for people's awareness of the importance of biodiversity?
Yeah. Okay. So the question was, referring to the large number of people, sort of funders and interest in biodiversity among all these different sort of organizations. I mean, I should say that this is I'm part of a much larger grant that has a multitude of sort of things that are being measured. So I can't necessarily say all of these people are funding this project because of their interest in bees. They might have, it's greenhouse gas mitigation is in the title. So they may have other rationale for it. But things like Ducks Unlimited, Nature Conservancy, obviously Genome Canada was actually sort of, and Genome Prairie was sort of very encouraging and sort of getting bee work into this project. So I think there is a lot of interest. I've had support from other organizations, so more directly on the work that we've been trying to do on bees. But no, I think people recognize that biodiversity is contributing. Sometimes people think of it as like economic, what do you call them, ecosystem services with kind of a financial or cultural sort of significance. I like biodiversity because it's fun. But no, I'm not answering your question very well, but I'm just kind of circling around it. But no, I think a lot of people recognize the benefits of biodiversity. And I think there is it's coming down the pipeline. I think that people are going to say, okay, greenhouse gas is a very important, but biodiversity is actually a really important thing that we need to pay attention to before we lose it, because it is going to mitigate all these negative things that we're trying to avoid.
Please.
Jason, you talked about biodiversity and all the various aspects of bee diversity. You didn't say very much about conservation specifically. Have you done very much? We looked at the species just in Manitoba, for example, and tried to rank these things about the risk levels and so forth?
Yeah, so the question is, I've talked about biodiversity, but I haven't talked about conservation and whether or not we've tried to rank species. Other people have done a number of, there's like kind of an IUCN kind of system of rankings. Oftentimes things are data deficient because we don't know enough. I haven't, explicitly been involved in that kind of rankings. There was one species that was treated as endangered by Costiwick, Epilotes pilosulus, and a couple of the students found that in Manitoba for the first time after 95 years. So we are engaged in that sort of sense. So we sort of share those data with organizations to say, actually, this bee is present. But I haven't gone to the effort of following through on the area covered or the occupancy to try to come up with formal assessments for all the bees. If anyone's interested in doing that, we have all the bees. So.
This question's a bit niche, but you talked about how it used to be thought that eusociality was kind of like an endpoint and there was no going back from it. Is there any evidence in bees or any other kind of group that there could be kind of a reversal of like social or group behaviors?
Yeah. So the question is, eusociality has reversed, but is there any evidence that brood parasitism has reversed? There isn't in any phylogenetic sense. There's no clade of brood parasites that we know that has a non-brood parasite nested within it. But it would be very hard to know, brood parasitism probably evolved from facultative kind of nest disruption in otherwise nest building species. So it's possible that there may have been populations at some point in evolutionary history that started doing the nest disruption thing, but then went back. But we don't have any data of something that's, you've lost all the pollen collecting apparatus and now you're nest building again. Occasionally I see the brood parasitic Lasioglossum with small amount. They have sometimes residual scopa, pollen collecting hairs, and every once in a while you'll see a little bit of pollen in there. So they may have some relictual behaviors, but.
I have one more question, if I may take another turn. So you have these bees that look very similar to the naked eye. You mentioned that already, particularly the same species group or genus. Is there, as an engineer, I'm always going to ask about technology, but is there technology, whether that's drone-based or otherwise, that you have to look at them under the microscope, obviously, but how powerful is new technology to differentiate between species?
Yeah, so the question is there other technologies for differentiating between species? There are people investigating this sorts of thing. So using kind of machine learning approaches where you basically train computers on images of bees to try to identify them. And I think it's still very much in the proof of concept stage. So there's a paper that came out recently that I was one of the reviewers for, and they had pretty good success at identifying sweat bees, but they had a very limited pool of species that they were working on. So they had like 10 species, and they're like, well, we trained it on these 10 species, and we did a pretty good job of identifying those 10 species. But really, if you go out and collect, you're actually working from a pool of 50 or 60 species. So I think that there just needs to be more effort going into building those data sets to train those, and maybe it would work. Because some people have done it on wings, just the wings, so characters that we don't, as a taxonomist, I don't normally use to identify at the species level, but people are finding evidence that you can do it, maybe. But I think it's one of those things where you need, there needs to be more interaction, because these are usually people who are operating off on their own, but you need the taxonomist to go in there and actually identify them as much as possible to get that pool, and then hopefully have this sort of iterative process of the machine says that you're wrong, and then that might teach the taxonomist how to do things better too.
So no crocodiles in Manitoba. So
no, well,
the same idea there.
Have we looked everywhere?
Yeah.
I don't know.
Pretty fine. Any other questions for Jason? Julia.
Well, do you know of any study or studies related to comparative genomics using sweat bees and different levels of sociality? And if there are any genes or pieces that are associated with that behavior?
So the question is there any comparative genomics work on sweat bees, trying to understand genes, kind of things associated with sociality? Yeah, they're genomes. So Karen Kapine, who's at Utah State University, and Sarah Coker, who's at Princeton, are two people who've worked on this kind of area. So there's an interesting group of sweat bees called Megalopta. They're nocturnal, kind of flying ones that occur in the neotropics, and they have both social and solitary nests, even within the same species. And so Karen's done a lot of work on that group. And then Sarah has done work in Lassia blossom. There's sort of similar things, species like that in Europe, Lassia blossom aldipes. But Sarah has some old school approaches, so she's actually trying to have nests in the lab so that they can study the biology of the nests while also doing the genomics work on top of that. So pretty cool stuff. So we're actually, there's a whole other project on trying to understand the social evolution of collected bees that Sarah and others and myself are kind of involved with.
Time for one last question, if anybody has one. Okay, seeing none, please join me in thanking Jason for his excellent presentation. And just go ahead.
Oh, thanks for the invite.
Oh, my pleasure. Just a reminder, this seminar series continues. We have speakers coming up, so keep an eye on our social media feeds, on our YouTube channel. Jason's presentation will be there as well. So if you have any colleagues or friends that couldn't come today but want to, learn more about this topic, please refer them to our YouTube channel and to our Make Manitoba website. So thanks everybody. Have a great evening. Thank you.